here. I'm just going to talk about the foundation of soil and plant health. Our developer used to say if you took care of the soil, the plant would take care of itself, and that's really what we're going to talk about here today. So we're going to do a little bit on histories of microbes. We're going to go into program, not product. Unlocking the potential of plant growth promoting rhizobacteria, a little bit into nitrogen fixation, phosphate solubilization, potassium mobilization, a little bit of the phylosphere organisms, and then we'll go into questions. So, how many people have heard of Dr. White? Okay, I'm going to refer to Dr. White a little bit in here, and
made these slides to kind of contrast what I call chemical agriculture, using mostly chemical inputs, with biological agriculture, regenerative agriculture, right? I call it biological agriculture because microbes are involved. And just to contrast, the products of chemical agriculture are generally less nutrient dense, and the crops themselves tend to be require more chemistry to control disease and so forth, because without microbes you get more disease that you have to then use more chemistry to control, and more fertilizer to push the crops, and more problems with stress. That's the other issue, because plants don't become stress tolerant without microbes. But with microbial agriculture, biological agriculture, there's a lot of recycling involved. Products are more nutrient dense. You don't get any runoff. In fact, it's more like closer to 100% efficiency, whereas conventional agriculture, fertilizer is probably about 30 to 40% efficient, with most of it being runoff. But you don't get any runoff in biological agriculture.
So, one of the things I want to talk about here today is basically where I come from. I really feel the perfect grower is the conventional grower who is biologically minded. He has all the tools in his toolbox that he needs conventionally if he ever needs to use them. But he takes a biological approach. Example would be if, we always have the debate, no-till versus strip-till versus tillage. Sometimes putting, I call iron into the soil, is one of the quickest ways to fix an existing problem. So to be 100% one versus the other doesn't always make the most sense, because sometimes you need to utilize those tools. And a lot of times what I say when we're doing biological agriculture is if we go into a field and we disrupt it, let's say we spray a herbicide, we till, whatever we may do, as long as we know what we've disrupted in that biological community and now go back and fix what we have broken, whether that be a food source, whether that be a cover crop, whatever that situation might be, to try and bring that biological community back up to the point it was before we went in and damaged it. So that's sometimes the way I look at it, because I always say, I love this quote from Paul Harvey: man, despite his artistic pretension, his sophistication, and his many accomplishment, he owes his existence to a 6-inch layer of topsoil and the fact that it rains. I mean, that's really what it comes down to. And the far side is always great. You little softies, when I was your age, I had to crawl through 14 inches of the surface and back. And I mean, to even see worms in agriculture today it's not the norm anymore. And to see topsoil of, 6 inches, 8 inches, 10 inches of actual true topsoil is, they do. The worms have it easy these days. So a little bit of information on biology, and I always go through this. I'll just go through it really quick because I don't think a lot of people understand this. The patent for rhizobia was done in 1896. So we start to talk about biological history and how long it's been around. 1953, the first time of liquid inoculums were used. In 1978, the term plant growth promoting rhizobacteria was coined. The first commercial application of inoculants was in 1982. 1953, mycorrhizae was used in strawberries, and proof positive of the effect of mycorrhizae fungi was in 1957. So we got to get out of the past. Biology has a function in agriculture today. I think that's why we're all here. If you were a drowning victim, the term you get smoke blowing up your backside, I'm not here to blow smoke up anywhere. But we need to give those bugs in a jug a hug. I kind of like that saying because we're referred to as bugs in a jug. The funny part about it is they're not bugs, but anyway, that's a whole nother story. So who is Tanio? Bruce Tanio was a seed geneticist for the Wilbur Ellis Company. He would create hybrid plants, basically. He would create a variety of wheat that was resistant to rust. 2 years down the road, that rust would mutate, and now suddenly that plant that he had bred is no longer resistant to rust. And so Bruce started thinking about that, and he's like, I am not creating healthier plants, I'm creating healthier insects and diseases. And so we started to look at why, and it came down to nutrition and how plants get nutrition. And you know, I call it, in agriculture sometimes, this is we're chasing rather than the carrot, the donut, because we'll never catch it trying to go through plant breeding, better varieties, without taking a look at how that the plants get their nutrition. Created down here, you can see this scale. And this was based on pH. This was in 1987. That if we had a plant pH of 6.4, and Bruce would say this doesn't matter whether it's a pecan tree, a tomato plant, a corn plant, coffee plant, if we pay attention to, this is tissue pH, not soil pH, tissue pH, that we can predict our susceptibility to insects and or disease based off of where that pH is of the plant. The other thing he tied it to is based off the nutrition. Is it an amino, a cation? What is the excess or the deficiency that is causing this susceptibility? And this chart here on the pH and the bricks is directly correlated to this pH chart. As we balance nutrition in the plant and we eliminate excesses, we increase our bricks level. So from our chewing, our aphids, our sucking insects, any of those types of things, if we can get our pH between a 6 and a 9, it's not food for them. And it's all how they go through, it's all how they see that plant as nutrition. Insects see infrared light. So when a plant is stressed, it puts out a different frequency than a plant that is not stressed. And that's basically how plants decide what they're going to feed on. I mean, they're there to clean up sick plants. That's why insect and disease exist. We're not going to get a whole lot into that today, but when we start to talk about biology, Bruce used to say everything in this universe is connected to everything else. You cannot isolate one mineral, one soil, one microbe, one plant, animal, or human component and treat that component as though the others don't exist once you get an understanding of the interrelated character of all things. And when we do agriculture, a lot of times I say it's like putting a puzzle together. It's finding that missing piece. Or it's the idea, if there's one person putting the puzzle together, it takes time. If you have 5 people putting the puzzle together, it goes much quicker. And that's sometimes what I look at when we start to talk about these things. If you go out and you put a cover crop in, and you go in and spray it with glyphosate to terminate it, and then you till it under so that you can go in and work your field, have you really made a change based off of soil health of putting that cover crop in? So did we take 1 step forward and 2 steps backwards by a herbicide application and a tilling? So the human component of what we did, of disrupting that biological community, could have been more detrimental to the biological thing that we tried to do that was good for our soil. So sometimes we have to have an idea of how everything is connected. And when I talk about that, I'm going to talk specifically about biology for the most part today. But when we think about biology, everybody talks about the rhizosphere surrounding the roots. That's what we talk about. That's where we focus on. We have to have an understand that it's the roots, the root surface. It's in the flower. It's in the fruit. It's in the stem. It's in the leaves. It's inside the plant tissue, and it's surrounding and inside our seeds. So when we plant a crop, where we get our seed from and how our seed is grown had a lot to do with basically biological expression of how that seed grows. So seed quality makes a difference. When we start to talk about You know, I talk to guys all the time, well, I'm doing a foliar fungicide, it's really not going to affect my biological community in the soil. Well, the biology is all over the plant. And we're going to talk a little bit about how it all functions together as we start to talk about it. So let's start with plant growth promoting rhizobacteria. We basically have the direct effects that we all talk about: nitrogen fixation, phosphate solubilization, potassium. We talk about nutrient acquisition, growth and development, and phytohormone production. Those are all part of plant growth promoting rhizobacteria around the root. And then we look at the indirect effects, antifungal properties. We look at systemic resistance. We talk about polysaccharide production. Austin was talking about when he had his camera out, and he was looking at some of that on those sand particles. And that's the thing I love about those sand particles. It's like, wow, that's free silica. But as we start to look, the tolerance to abiotic stress. Now, abiotic stress can be drought. We've talked a lot about sodium today. Sodium stress. He talked about 14 days longer on his beans than his neighbor got. All of this is part of that biological community within that soil environment. So when I talk about nutrition and plant health and biology and how all these things are connected, this is what we have to think about. It is a living skin. The rhizosphere is a living skin. Plants, crops, people are all dependent on 5 favorable conditions. They need light, heat, air, nutrient, and water. With the exception of light, the soil can supply every one of those. You talked about his soil temperature being higher because of his biological community. We talk about building polysaccharides, soil binding particles. We talk about nutrients, but only when all of these are in a balanced combination can we get good overall growth and development. We always go over here, the law of minimum, but before we even get here, we have to start here, because this is where it all functions within that soil environment. So the law of minimum, basically, we always used to say deficiencies. If we know our deficiency, that's our greatest limiting factor. Actually, our greatest limiting factor is our excess. We talked a lot today, Todd talked about going out and putting more potassium, more calcium. What he's talking about is we are creating excesses within that soil that are blocking or causing a deficiency of something else because of an excess within that soil environment. And we can't, if we have too much mag in our soil, we talk about mag, tight soils, compaction, no air. We talked about microbes need air. If we talk about excessive sodium, we can't have good utilization of water. All of these things are interconnected, so these have to be as much in balance as this has to be in balance in order for us to reach optimum plant health and soil health. So how do we get there? You know, I look at it, we've always said test, don't guess. We've talked a little bit, we have to test our soil, our plant, our water, our products, our compost. Anything that goes on our field needs to be tested. You know, we do a soil analysis and plant sap analysis. And part of this is what we talked about. Here's what's in our soil. This is telling me what's available, okay? So there's a lot of times where you'll get a recommendation that you guys need to put out potassium. Well, we may have sufficient potassium and our plant's telling us we have an excess of potassium. So that's the last thing we want to do is go put more of what the plant doesn't want. So unless we have this information, we can't make a good decision. And the reason I bring this up is because the biological community runs on soil health and plant health. If we have excesses, our plants aren't sharing those sugars that we talk about. We'll talk about a little bit further as we go along. Because all of these things have to be in balance in order to feed that biological community within that soil environment. You know, I look at compost, for an example. Everybody says, well, I'm putting out compost because I want the nitrogen. But then suddenly you look at the sodium level and you're putting out 2 tons of compost to your field and you're actually putting out 25 or 30 pounds of sodium where you already have a sodium because it's coming from your irrigation water. So we think we're doing something good, but it's actually, it's detrimental to not only our soil health, our plant health, but also our biological community because we are what we eat. Basically, If we don't test, we cannot have good, healthy crops. And like I said, we also do biological testing. Biomakers, I don't know if anybody's heard of biomakers. There's a couple other labs out there that are doing DNA sequencing, which will tell you, they don't tell you the microbial strains within your soil environment, but they'll tell you who's there based off of function. So it's actually kind of a really cool tool when you start to look at it. The reason this is important is this is how nature works. This is, I again mentioned Dr. White. How many people are familiar with the rhizophagy cycle? Okay, so I'm just a couple, so I'm going to explain it. This is really cool. Plant roots are not great at extracting nutrition from the soil. In fact, they're terrible at it. They need these biological communities. So this plant will put out these exudates and recruit this biological community that we see down here. Well, at the root meristem, this bacteria and fungal will enter into the root. That's what you see all these little pink and blue lines. Inside there, the plant does something a little bit not nice, is it bombards it with reactive oxygen. And what that does is it makes those microbes swell up and become leaky. So it'll take all the nutrition out of that beneficial bacterial fungal community that's inside this plant, takes all that nutrition out, and then it'll spit them back out into the soil environment through its root hairs. The cool part about this is now they have to reform their walls. They're like a bear coming out of hibernation. They're hungry. So they get everything they want in this environment, and the process starts all over again. The cool part about this is Dr. White's research. Root formation is regulated by this whole process. All these fine root hairs are put on the plant specifically for spitting this biological community back into the soil environment. But it even gets better than that. These micropotaplasts are replicated within the cell root. So inside that cell, in here, if this plant brings in some phosphate solubilizers and it likes them, it will now clone them and spit out 4 or 5 of those into the soil environment. And all of these root hairs are stimulated because of that whole system. So down here you see this corn plant. This is a corn plant in Kansas. Whenever I dig up a plant like this and I see all these roots and I see all these fine root hairs, I know we've got good biological function because we're cycling through the rhizophagy cycle all of that nutrition into the plant. And so that's what we talk about, building soil health from the plant out. These are some roots, they basically in a dryland area of Washington. The funniest part about this, we were in a severe drought. The grower could not figure out where these plants were getting moisture from. You dug up these plants and they just had what I call the dreadlocks or the roster roots. All of that is built around that microbial community to build a home. They basically create this through their exopolysaccharides. They build these soil binding particles to create an environment for them to live. Okay? So inside there, they create this environment. The pH around this root structure right here is right around balance, 6.4, 6.5. That's the environment that they want to be at. So we talked a little bit, Todd was talking a little bit about pH and stuff. A lot of times we're never going to change our existing soil pH. It is what it is. We can dump all the products out there that we want to try and adjust pH, and all we do is kind of go back to what I talked about. We yo-yo the nutrition. We throw everything out of balance based off of trying to fix one thing, which is really something that we're really never going to change. Water is probably in irrigation is the biggest thing that is going to change your pH. Whatever your water pH is, that's, I mean, how many gallons, thousands of gallons of water are you putting onto your soil? That's going to dictate where your pH is. And most of the time I don't care because this biological community is going to adjust for it. This was here, actually some stuff that Wyatt, you had done with a seed treatment. And when we start to look at this and we now think of what we talk about on the rhizophagy cycle, and we see these dreadlocks. This was a, what, 0.9 organic matter. All of this is being built by those microbial communities within that soil environment because they are created an environment where they want to live because they're making nutrition available to that plant. That plant is sharing its exudates with the microbes because basically they support each other within that soil environment.
Because what would have happened in the real world is you would have had microbes from internal, and you would have had things immediately coming from outside. In fact, some of the things from outside even have an effect on the germination rate of the seed, improving germination rate. So we know that they're influencing early germination.
So when we start to talk about that, a lot of times I like seed treatments to get the seed off and going. When that seed germinates and that plant starts to grow, that seed will share up to 75% of its sugars with the microbial community within that soil environment to build this right here, because that's the fertilizer factory for that plant. It knows if it gets this microbial community up and functioning, it's going to get all the nutrition it needs in a state that the plant can use it, as long as we don't do anything to disrupt that whole environment. So until we get that whole thing functioning, I guess this is sometimes what I say. In agriculture, a lot of times we say we put out NPK and we put out these special products. Adding vitamins to donuts does not give you pep and vigor. It's the sugar in the donut that's really getting you going. So a lot of times on that premise, this is what we express: a nutritionally deficient plant, because we are trying to chemically feed that plant something that it does not want. I was in Europe last year. I had to go from France to Portugal. This is the freight train system. So if I wanted to go from France to Portugal, I had one way to get there. That's kind of what I look at when I see roots under our general agricultural practices. We basically are missing most of the countryside. So if I wanted a good bottle of wine, I'm limited to what is on this. If I wanted a bottle of port from somewhere in Portugal, basically I get it right here. Where we look at the biological system, this is a commuter train system in Europe. I can go anywhere I want in that country and see any part of that country, get anything. The same holds true when we develop the roots that we see like this. We suddenly have the rhizophagy cycle, nutrition being watered. We have water available. We're starting to build soil health, carbon, polysaccharides, glomalin. All of these things are starting to be built within that soil environment. Because remember I said we needed air, nutrient, water in order for this microbial community to be in balance and live. That's the same thing we need to make nutrient availability. These extra polysaccharides are exactly what's doing it. They're the soil binding particles which create soil structure. And so when we think about building soil structure here or building soil structure up here, which is going to build soil structure quicker within our agriculture environment, where we have more contact with that soil particles, with those exudates, with those polysaccharides going into that soil binding particle. Because what it comes down to, I love to fly fish. This is me in Colorado because I don't like to fly fish with other people. So this was January in Colorado. It was like 23 degrees outside, and I'm standing in the river fly fishing. The reason I tell you, this section of river that I'm fishing in right here, 20 years ago, there wasn't a single fish alive in this river because of chemical contamination, lack of habitat, nothing would support fish. Today, this is a gold medal water because they went in there and they reduced the contaminants going into this part of water. Suddenly, we had biological life, we had insects, we had all these beneficials because the habitat for biodiversity can only be there if it will support it. I always say that environment dictates expression. So, you know, we talk about primary succession. We talk about bare rock, the lichens show up, then we start to see the bacteria show up, and then we get up here to bacteria and fungal, and then here's more fungal dominant than it is bacteria. In agriculture, we're moving backwards a lot of times rather than forward. And when we start to talk about this, we were just talking about weeds, based off of weed expression, based off of soil structure. So we're going to have different weeds here than we are here. You had mentioned amaranthus. Yeah, it's non-mycorrhizae plant. That's when they start to show up. I look at that from that standpoint, and we've worked sometimes here rather than up towards here. You know, we can see that on all the weeds and how they express themselves within the soil environment. Cheatgrass in wheat. I had a grower that we were working with, and it was a real bad year for cheatgrass 2 years ago, and I went out and asked the grower, I said, did you guys spray this field with cheatgrass? I thought we agreed we weren't going to do anything on this field. And he goes, no, I didn't spray it. And I said, cheatgrass is really bad. And he goes, this used to be my worst field for cheatgrass. He said, since I started doing biological principles, and it wasn't just biology, it was biological principles, he said, I no longer have cheatgrass in this field. He said, it has changed now. He says, I have other weeds, but the cheatgrass is no longer a problem. And a lot of that is because of primary succession. We have now moved that soil to the next level, which I kind of get really excited about. So anyway, how much time do I have? As we look through here, what I always say that doing this biologic process, it takes time. We have to be patient. All the processes that we can build upon each other are going to have it happen quicker, all right? The changes build upon each other year after year, and the changes are not only in the plant and the soil, but they have to be in you as a grower in order for them to be successful.
35 minutes.
How many?
35.
Okay. So how do we make those changes? First thing I'm going to talk about is nitrogen, because it's a low-hanging fruit. There's more to it than that, but excessive nitrogen shuts down the rhizophagy cycle. So what we just talked about based off of nutrition is totally shut down when we start to talk about the rhizophagy cycle. When we start to talk about insect and disease and susceptibility to insect disease, it is all directly correlated to excessive nitrate nitrogen. There was, for every 2 kilograms of nitrogen that is put out into the soil, that is in excess of what the plant needs, and that's going to be crop dependent, depending on what crop we're growing or how much nitrogen we use, will burn up somewhere around 200 kilograms of organic matter or carbon. And when we start to think about that, is if we're trying to build stable carbon in our soil environments, we're trying to grow basically an environment, organic matter, to hold water. What is it? 1% of organic matter holds 27,000 gallons of water. Now we start to talk about Todd's water, and we start to have holding capabilities and be able to retain that within our environments. I mean, that really starts to build upon itself. But when we start to think about that, nitrogen is a big culprit and probably is one of the ones that's most used. I've heard a lot, but talk about the moron principle. Yeah, I usually call it is the moron principle. We're going to put more on, which doesn't make a whole lot of sense. So this is some data based out of a wheat trial, variety trial that we did this year in Washington State. This was in the Palouse. We did 13 different varieties of wheat because, again, based off of breeding, a lot of times the breeding for genetics of wheat is for high nitrogen utilization, so they express themselves used under high nitrogen. So we wanted to prove that we could do it biologically. So we had 120 pounds was a standard check. 120 pounds matter. Remember, I just said 2 pounds over the needed nitrogen burns up 200 kilograms of carbon. I mean, what was the cost to grow 5 more bushel of wheat? It's not always there. But the really interesting part of this is what I love. So this is a plant sap from apical. This is the control, and this was the treated. On the treated side, our nitrogen, total nitrogen, was higher with 25 pounds of nitrogen than it was with 120 pounds of nitrogen. Our potassium was higher, our magnesium was higher, our calcium was higher, our manganese, cobalt, phosphorus, silicon, iodine, boron, molybdenum, and selenium were all higher in the biologically treated with less nitrogen. Now, part of that goes back to the idea of the rhizophagy cycle. Basically them taking the microbe in, digesting that microbe, and utilizing that for food. But I'll go ahead and play this.
pH on those two? Out of curiosity.
What's that?
What was the pH on those two?
So the tissue pH
Didn't come through.
did not come through. Reason for that is we did not have enough tissue to do the full tissue pH and everything. When we were utilizing these trials, that was the one thing that we had to be careful of, because the other thing that I did here is, in addition to doing, and I didn't have time to put it in here, the biomaker testing. Biomakers testing, resiliency was much higher, which is how adaptable is the crop to stress, really is what it comes down to. And basically everything that we see here based off of nutrition showed up on the biomakers test as showing up also as better than the control. But we were pulling plant saps every 2 weeks, and we realized there was going to be no tissue on the places that we were sampling if we kept taking it all. So we reduced the amount of tissue that we took. So
Find that in other experiments, when we actually look at plants where we suppress superoxide and measure nutrients, compared to those where we suppress superoxide and measure nutrients, then what we find is the divalent metals are really, really significant in the oxidative portion. Those divalent metals, things like magnesium and iron and calcium and that, manganese, and so forth. Like zinc, and all of these nutrients are impacted, but these divalent metals tend to be the most significantly impacted. Manganese is really special for rhizophagus cycle, like 700% more compared to without, very much more manganese. But these are really important nutrients. So plants are using rhizophagus cycle to get the most critical nutrients for their development.
So a lot of times get asked, well, if you only used 25 pounds of nitrogen, where is all the nitrogen coming from? And I think we talked a little bit about that today. Free living nitrogen fixers. I mean, just like we know rhizobia fix nitrogen on our legumes, we have free living nitrogens within that soil environment. Basically, there is enough atmospheric nitrogen out there to support these crops. I want to say,
78%.
Huh?
78%.
78%, yeah. Comes, even when you're under a conventional program and you're using conventional nitrogen, the report came out that still 78% of the nitrogen came from biological, 67 parts, sorry. 67 parts came from biological nitrogen. So these are free living nitrogen fixers. If we use excessive nitrogen in our environment, these free living nitrogen fixers do not have to do their job. They're lazy. If the plant doesn't need nitrogen, why am I going to do my job? But we only have free living, we also have endophytic nitrogen fixing bacteria. Remember I talked about around the root, in the stem, in the leaf, in the trichomes. All of those places are going to give us nitrogen fixation within the plant. I'd like to talk more about this, but I'm way behind schedule here. So we start to talk about rhizobia. We all know rhizobia and fixing nitrogen, our legume plants. Co-inoculation with phosphate solubilizing bacteria has shown a 31% increase in nitrogen fixation. So if we're planting any legume, we have to pay attention to our phosphorus levels. If not, a lot of times a phosphorus inoculum may help get you to fix nitrogen. A lot of times I'll go out there and say, yeah, you know, I've got my nitrogen fixing crop out there, but I really don't have a whole lot of nodulation. And a lot of times we talk about nickel, molybdenum, cobalt, all cofactors of helping with that. But a lot of times it comes down to phosphorus, because nitrogen fixation takes a lot of energy, and the battery that gives us that energy is phosphorus. So this was a trial we did with, this was garbs or chickpeas. Sorry, it was a joke. Never mind. Most people don't get it if they don't know that chickpeas and garbs are the same thing. But here you can see on the left, we're untreated, and on the right, we're treated. This is a little bit better picture. The thing I got excited about when the farmer and I went out and dug this up was all the nodulation that we saw on this side, and there wasn't really any. That's the chickpea. That's not nodulation. What the grower got excited about was the root mass, because he said, I can grow great garbs, I can fill pods, but what happens is I never have enough moisture to fill all my pods and go to harvest. So he said, you know, I may have the potential for a great crop, but I abort half the pods because I ran out of moisture. So he was excited about that. So when it came down to it at harvest, the treated with the inoculum basically, he doesn't care about yield. He only talks ROI. Yielded him $753 basically more per acre. The net on the control was 432. On this side here, this was a treated, but it also got a foliar application. Now, had we not got hot and dry and ran out of moisture, this would have been the highest yielding, but because of the abortion of the pod, because we did not have the moisture in order to take them to finish those plants off, we were down at $587. But a $34 investment yielded $753.49 per acre. That is just a phosphate solubilizing bacteria. That's the only thing that was done differently there.
Technical question, but in your experiments, are you starting to understand how the plant communicates to these bacteria? You mentioned that they will actually leave with some type of signal to go retrieve a nutrient to bring it back into the plant. Do we understand how that is actually happening?
Yeah, that's not complicated. It's not complicated. It really is quite simple. At least in terms of nitrogen, if you think about it, plants lack nitrogen. What they do is they withdraw nitrogen from their exudates. Don't put out any nitrogen. Instead, they put a bunch of sugar out and organic acids and so forth, but no nitrogen. And what that means is that the bacteria who are able to grow there, replicate and grow there, are the ones that are able to fix nitrogen. Those are the ones they take in. There's plants are manipulated for other nutrients. There's other, they have other strategies for other nutrients, which is not as easy to understand. And so I'm not going to talk about it. But the nitrogen one is very easy to understand. And, that's how that works.
So, that kind of comes down what I was talking about, the excessive nitrogen. If we have excessive nitrogen, the nitrogen fixers don't do their job. They don't have to. So when we start to talk about plant communication, he talked about how do the microbes communicate basically needing nitrogen. Well, it's not only nitrogen. We sometimes talk about root exudates as being sugars. That's kind of the simple way to state it. Plant can select their microbiota or microbes by releasing root exudates. Microbes are attracted by chemotaxis to the rhizosphere by these secondary metabolites, low molecular weights, amino acids, organic acids, sugars, and high molecular weights, compounds, polysaccharides, mucilage, proteins. But this is a direct communication that the plant spits out through those root hairs to specifically recruit specific microorganisms within that soil environment. So over here on this side, we look at our basically our stress tolerant to our biotic stresses and our abiotic stresses. Over here are our disease pathogens. Here's what we talk about. Disease, insect, those types of things are all on the biotic side. The abiotic side would be drought stress, sodium stress, the things that we talk about. They will recruit specific organisms, kind of like here you see this picture of all this green surrounding this root. This is Bacillus subtilis. This plant was under attack by a disease, and it recruited these guys in order to protect it and surround it and protect it from the disease pathogen. So when we start to talk specifically about plant stress, this is one of the things that I kind of want to point out here as we're trying to build soil health and we have high sodium levels. So under a non-biological program based off of exudates, we increase the amount of sodium in the plant under stress, and we reduce the amount of potassium, okay? Under a biological, we do the exact opposite. We remove the sodium from the plant, we take it from the shoot back down to the root, and we put it in the potassium. The reason I say potassium is critical, potassium regulates the opening and closing of the stomata. So if we have a potassium deficiency, that plant can't shut itself down during high heat periods of the day. So they stay open, it basically loses more moisture. The other part about this is really cool is it says, I have a really good graph that shows all this, but I knew we weren't going to have time. But it says it moves it from the shoot to the root. So the plant can take, with the communication through these biological, they can take sodium out of the plant, put it back down here into the soil profile, as you can see right here, and then the biology will tie it up with polysaccharides within that soil environment. We're not getting rid of it. They just tie it up and make it not plant available. That's, as I call it, that's a Band-Aid. It helps us to grow the plant under these high sodium environments, but a lot of times we have to stop the reason that we're putting sodium out, whatever that might be. If it's our fertilizer, if it's our irrigation, if it's our water, whatever that is, we have to fix the problem, and we can't just keep dumping it within our soil environment. But we see a lot of different water balance and maintenance, antioxidant production, extracellular polysaccharide enzyme productions all happen within this. So we're going to move on to the phytosphere. I don't think I have much time left. I love this picture because everything that happens basically in the rhizosphere is exactly the same on the phytosphere. So if we want to start talking about colonizing microbes play a critical role on multiple functions, including productivity, fitness, leaf function, longevity, seed mass, apical growth, flowering, fruit development, are all key parts of basically the phytosphere of the plant. So when we don't think about the phytosphere being tied to the rhizosphere, we're making a huge mistake when we start to talk about overall biological health. Because everything you see here, pathogen warning, we see that comes up into the plant, down into the soil, basically into the soil and back into the plant. There's a direct communication across here on all the different parts of the plants. Basically, whether we're talking direct pathogen communication, whether we're talking function of nutrient availability, immune responses, water uptake, nutrient availability, all of this is a communication pathway for the plant. And when we start to talk about this, it's not only on the leaf structure of the nutrient roles of the plant, climate regulation, we talk about pollution control, we talk about bioremediation. All of these things happen on the plant and within the soil environment also. This is kind of cool. This just talks about the research of phosphate and potassium mobilization. We talk about iron, zinc, sulfur, copper, manganese. All of these are part of the system of making nutrient available to the plant. And again, it goes back down to building that foundation. The more we can have that plant photosynthesizing, the more efficiently it's working, the more exudates it puts into that soil environment to feed that microbial community, the more of the rhizophagy cycle we see get going on. This is kind of cool. Biology actually creates a biosurfactant function on the leaf of the plant. So when we add biology to our foliar applications and we pay attention to our water, water is a big part of foliar applications. I didn't touch on that a whole lot, but if we have poor quality water with high bicarbonates and we add foliar nutrition to that, we're tying most of that up before it ever gets to the plant. So water quality is a huge part of our foliar applications. We have to understand that. But they create this biosurfactant. The reason the biology does this is because it needs basically water. It needs that. It doesn't do it to help get nutrition into the plant. It does it for its own survival. But it's kind of cool when the research is coming out there of how biology creates this wettable area on the leaf of the plant. And we know if we're going to do foliar applications and we want to get good nutrient uptake, that has to stay moist on the plant. Once it dries out on the surface of the plant, we shut down the utilization of that foliar application. And this is what I think is really cool on the phytosphere of the plant. How many guys have heard of quorum sensing? Okay, so you basically, quorum sensing is you get a group of microbes and they put out these volatile organic compounds that say, man, the environment is right for us. Come on guys, everybody come here, we're gonna get together and we're gonna attack this plant. Well, when you have organisms on the phytosphere that are beneficial, they do quorum quenching. So when these guys do their quorum sensing to get them together to attack the plant, these guys are over there eating up all of their volatile organic compounds and basically suppressing the signal so that they can't group together in order to attack the plant. I mean, I think that's pretty cool stuff. Sorry. I love this kind of stuff. Here's going to kind of show you. This has no sound, so I'm going to have to kind of point it out to you. This is really cool. So see all these greens? These are all disease in an untreated plant. And right here is that little green opening of the stomata. See those little green guys going inside there? Can we see that? Yeah. Okay. Yeah. So this is a treated plant. You can see the red glow. Basically, they've shut that stomata, they've shut that plant down, and they said, you're under attack. Don't let these guys get in. You can see this guy over here trying to get inside the plant. He's not making it because there's no openings. The cool part about that, we were talking about calcium. We were talking about silica. Somebody mentioned silica today. I don't remember who it was. All of these are critical to cell development of the plant. So when we start to talk about some of our diseases, biology alone isn't a silver bullet. We have to have good nutrition in order to get good hardy cell development. So when we start to talk about cell wall structure, that's calcium, silica. We need all of those in that plant to help strengthen that cell wall, to reduce the number of openings for all of these diseases basically to get in. Because these are all the listed EPA biopesticides. I say when you go to a point that you're using a biopesticide in order to stop a disease, you've already lost yield. You're already under attack, you're already compromised your plant, you've already reduced your income. A lot of times what I say is let's utilize the guys out there to keep this from happening based off of good soil health, good plant health, good phytosphere, good nutrition. So this is just some of the different research that's out there, but I wanted to share this with you because Fusarium, when we start to look at Fusarium as a disease, these are all of the organisms directly correlated with basically reducing susceptibility to Fusarium.
15.
What's that?
15.
Yeah. Till questions or 15 minutes left?
Till done.
Okay, so I'm done. Okay. Phytophthora, basically. Powdery mildew. Again, powdery mildew is one of those. These guys are great, but unless you have cell integrity of the plant, you do not have a deficiency of calcium, you're not expressing trace mineral deficiencies, powdery mildew is a really tough one to come after. Rhizoctonia, verticillium, and the list goes on. This is kind of cool. This is mycorrhizae here. Basically create little lassos and get your nematodes and then choke them down. This is an insect larva within there. Basically being attacked basically within that soil. Basically, chitin is part of the structure of this little guy. So chitinase enzymes will break this guy down. Sorry, fun stuff.
That results in hardiness, increased hardiness and stress tolerance in plants. And that means their climate change, it means salt, it means heat, it means diseases. The plant gets tougher and hardier. Also soil pathogens, the microbes that are involved in rhizophagy cycle come out of the plants, and some of them will colonize pathogens, fungi in the soil that cause disease, things like damping off pathogens, alternaria, for example, and fusarium get colonized, and the bacteria that go all over the surface but also go inside the hyphae, and that changes the behavior of the fungus. Fungi tend to be not aggressive, not pathogenic when they have bacteria.
So I kind of say it's kind of like sending out the bat signal. When the plant's under attack, sends out the bat signal, you bring in those guys and they give you that pow, that bam. But pest and disease do not arise because of the deficiencies of pesticides in our environments. They are symptoms of poorly nourished soils and crops. And that's really what it comes down to. I have to read this one. This was one of my favorite. Agriculture is the oldest of all industries and one of the most complex and difficult to master. Farming requires more intelligence than any other industry or occupation. The work is with living things, and living things cannot be managed by coercion. They must be understood. I mean, that really comes down to it. And then the rest of it, basically, the farmer must be keen, observant. His whole livelihood depends on a daily and hourly of his keenness and faithfulness of his observation of 1,000 things that the town people are utterly blind to. I mean, everybody thinks that the food just shows up on the shelf of the grocery store. They don't see the blood, the sweat, the tears, the pain, the hours that go into to make it happen. So with that, we'll go to questions. Sorry. Any questions? Wow, no biological questions. All right, thank you very much.
Dennis, I think we have one question up front. Right here, Dennis. Right here.
Oh,
You talked about excesses and the connection between nitrogen fixation on the root of a legume, for instance, being connected, the lack of fixation is connected to a nitrogen excess. And so, do you have to start putting less nitrogen on in order to start seeing fixation?
yeah, so...
More to it than just that. Talk about that.
Yeah, so a lot of times what I look at, and that's a really good question. Thanks for asking that. So biology is not great at fixing nitrogen in cooler temperatures. So when we have damp, wet soil, biology is not great at fixing nitrogen. It's kind of like phosphate solubilization or phosphorus availability. Without phosphate solubilization in those cooler temperatures, you don't get phosphorus available. But with phosphate solubilization, you'll see an uptake of phosphorus. Nitrogen is the exact opposite. Those nitrogen fixing guys do not work in cooler temperatures. So we have to have enough nitrogen out there to get that plant up and growing a lot of times early on in the spring. For example, in that wheat trial, we did 10 pounds of nitrogen. That's all we did. They did 120 pounds, or in the fall. They did 120 pounds in the fall, we did 10. That's all we did. And then we did more in the springtime, 15 more pounds. But form of nitrogen is really critical when we start to talk about nitrogen fixation. So any of your synthetic nitrogens, your salt-based fertilizers, shut down the rhizophagy cycle. So if we're looking at using nitrogen sources, a lot of time your corn steep, your soy proteins, any of those forms of nitrogen are much better nitrogen sources to utilize than a synthetic source. Sometimes you have to use some of the synthetic, but throw the other in with it as more of a biological catalyst is what I recommend. So, you know, example out here, you know, if they're doing cotton or whatever on your, you know, for your pecan trees, you can throw a little bit of synthetic nitrogen out there, but make sure there's an amino form or a soy protein form, something that the biology actually likes. And then that will not have as much of effect on the nitrogen fixation process. On the rhizobia, so much, most people aren't putting a whole lot of nitrogen out anyway when they're doing a legume crop. So a lot of times more what I look at is the phosphate solubilization, making sure there's enough phosphorus there to fix the nitrogen. Did that answer your question? Okay.
I have a question. When it comes to pathogens, plant pathogens, so you talked about quorum sensing, quorum quenching, and I have heard it put forth that, and I've never read the research on it, but I've heard from people that have that there's been research done, and I'm just wanting to see if this is accurate, okay? Is what can happen if you have a field that has a known pathogen, okay? If we get our beneficial biologicals high enough, part of the function of suppressing that pathogen is that they actually go into quorum sensing mode where they're putting off VOCs that make the pathogen actually shut off gene sequences to change its metabolism. So the gene sequences that express are not plant pathogenic metabolism. Is that true?
Correct. So there are a lot of fungal diseases out there that when we have the right balance of beneficial bacteria in that soil environment, they will infect that fungal, and now rather than that fungal being pathogenic, it is now beneficial. They basically change its whole process within that soil function. All microbes, all fungals have multiple functions within that soil environment. What they do is being expressed by nature or the plant. That's what's going to dictate what their main function is. So by having a balanced biological system, you're correct. Trying to think of the word I want to use. that disease pathogen now, that fungal pathogen now becomes, either it just shuts down and it does something else, or it could actually become beneficial to the crop that is being raised.
And so, if that's true, then trying to eliminate that pathogen from the soil is pretty much futile.
Well, I guess my question would be, in the history of fungicides that have been on the market today, how many pathogens have we eliminated out of the soil? Just give me one. Does anybody have one? They don't exist. And in fact, they've gotten worse. And going back to what Bruce talked about, is now what used to control a fungal pathogen with one mode of action takes 3 modes of actions, and rather than 1 application, takes 5 applications in order to get the exact same response that we had before. And now we're having to change modes of action in order to go after it. And so that's where, and it goes well beyond, you know, sometimes I will be the first to say, everybody gives biology too much credit. I mean, like those biological fungicides that I posted. Well, if I get whatever disease it might be, I can go out and spray this biological fungicide. By that point, we've already missed the boat. It's harder to control once it's expressed itself, and we're just shutting it down. We're not stopping it, let alone the cost that it's taken based off of overall yield that we have on that plant. And the other thing about it is, those organisms, if you test any soil, you can find pathogens in your soil at any given point in time. We never get rid of them. They're always there, okay? Whether they express themselves is another story. Again, I say it all the time, environment dictates expression. That pathogen is only going to express itself if the environment is right for it to do so. So,
Thank you.
Yeah. Anybody else?
We talked about a little bit about the refractometer.
Oh.
And I guess you didn't touch up on that. Can you?
Yeah, so we had mentioned that bricks chart up there and testing with the refractometer of your bricks level of your plant. So bricks, the refractometer is a great tool, and a lot of times you can utilize it, I say, with foliar nutrition. For an example, if you go out and put out a foliar application and you're used to using a refractometer to show soil or plant health, and you put out that foliar, and if the bricks level goes up, you know that foliar is what the plant wanted. If the bricks level goes down, you know that is, you did something wrong. That is not creating better plant health. And so with the refractometer, what I always tell you, it has to be done early in the morning. It has to be done at the same time of day. It has to be done on a regular basis. You have to eliminate any kind of stresses out of it. If the plant is stressed, you're not going to get an accurate reading. In fact, a lot of times you want to get bricks levels up in grapes, sugars up in grapes, shut off the water, stress them. It'll go through the roof. That's not a healthy bricks level, okay? So you have to understand that tool and how to use that tool. We always talk about bricks as being sugars. It's not. It's actually dissolved solids. That's really what a bricks reading is. So we're looking at nutritional density within the plant is a lot of times the way I look at it. Yes, it's sugars, but it goes well beyond that. So what we're trying to look at is how balanced is that plant. And the higher we can get that up, the more, I call it nutritionally balanced, we're going to have that plant.